Digital microfluidics is a technology that manipulates tiny droplets of liquid on an array of electrodes. It enables precise control over droplet movement without the need for physical channels. This reconfigurable platform allows for the automation of multiple laboratory tasks on a single device, integrating electrical, mechanical, and chemical principles to perform complex processes with high accuracy. It is transforming lab automation by making workflows faster, more efficient, and highly customizable. A digital microfluidic device can be programmed to mix reagents, incubate reactions, and analyze samples all on one chip, reducing manual intervention. Imagine a smart, modular kitchen countertop where you can reconfigure different cooking stations at the touch of a button to prepare a complete meal automatically. Droplet manipulation in digital microfluidics is achieved by applying electrical signals to an array of electrodes. This method controls the movement, merging, splitting, and mixing of discrete liquid droplets by leveraging electro-wetting, where electrical forces change the wetting properties of a surface. Precise control over each droplet is obtained by sequentially activating specific electrodes, enabling complex fluidic operations to be performed with minimal sample volumes. In a lab setting, a technician uses a digital microfluidic platform to merge two droplets containing different chemicals, triggering a reaction that can be monitored in real time. Think of it as playing a digital game where each move directs a small ball along a grid, combining with other balls to achieve a specific goal. Digital microfluidic systems are highly reconfigurable, allowing for dynamic changes in workflow without physical modifications to the device. These platforms are programmable through software, enabling automated execution of various laboratory protocols. The flexibility of these devices means they can be quickly adapted to perform different tasks as research needs evolve, with reconfiguration achieved by altering the electrical patterns applied to the electrodes. Researchers can update the software on a digital microfluidic chip to switch from performing a chemical synthesis to executing a diagnostic test without replacing any hardware components. It's like having a multi-tool that, with just a simple adjustment, can transform from a screwdriver to a pair of pliers, meeting different needs on the fly. Digital microfluidics automates complex laboratory workflows by integrating various steps such as mixing, incubation, and analysis on one platform. The automation minimizes human error and speeds up experimental processes, with sequential and parallel operations programmed to occur without manual intervention. This integration reduces the need for bulky, multi-dependent laboratory setups, thereby enhancing reproducibility and efficiency in experimental procedures. A clinical laboratory uses a digital microfluidic device to automatically process patient samples, performing steps from sample preparation to analysis, significantly reducing turnaround time. Imagine an assembly line where each station automatically performs its task with precision, resulting in a finished product without requiring constant manual oversight. Digital microfluidics significantly reduces the volume of reagents and samples needed for experiments due to its precise droplet control. This reduction in sample usage not only cuts costs, but also minimizes waste, while the technology allows for high-throughput testing by handling multiple reactions concurrently on a single chip. Efficient resource utilization is critical in both research and clinical settings, where reagents can be expensive or limited, making the technology both economically and environmentally advantageous. In drug development, a digital microfluidic platform can conduct thousands of microreactions simultaneously while using only microliters of expensive compounds, thereby conserving valuable resources. It is like a precision chef who uses just the right amount of ingredients to prepare several dishes, ensuring nothing is wasted and every dish is perfectly balanced. Digital microfluidic platforms are often integrated with sensing and detection systems to monitor reactions in real time. This integration can include optical, electrical, or chemical sensors that provide immediate feedback on experimental conditions, allowing for dynamic adjustments and enhanced overall process control. The synergy between droplet manipulation and detection makes the platform a powerful tool for diagnostic and research applications, and it also facilitates the automation of decision-making processes based on sensor data. A digital microfluidic chip equipped with fluorescent sensors detects changes in light emission as droplets undergo a chemical reaction, providing instant data on reaction progress. Think of it as a smart weather station that not only measures temperature and humidity in real time, but also adjusts your home's climate control system automatically based on those readings. Digital microfluidics has broad applications in both biological and chemical analyses, ranging from DNA amplification to enzyme assays. The platform's ability to precisely manipulate droplets allows for the execution of complex assays with high sensitivity and specificity, making it particularly useful in genomics, proteomics, and diagnostics, where small sample volumes and rapid processing are crucial. Its versatility enables simultaneous, multiple analyses, increasing throughput and data quality, and driving innovations in personalized medicine and high-throughput screening. In a research laboratory, digital microfluidic devices are used to perform PCR, polymerase chain reaction, assays on minute DNA samples, accelerating genetic testing and pathogen detection. It is like a state-of-the-art workshop where a single, highly versatile tool can perform various tasks, from precision cutting to intricate assembly, streamlining the entire production process. Despite its many advantages, digital microfluidics faces several challenges, including issues with droplet evaporation and electrode fouling. Maintaining precise control over droplet movement under varying environmental conditions can be complex, and the integration of multiple functions on a single chip sometimes leads to design trade-offs between complexity and reliability. Ensuring consistent performance and scalability for commercial applications remains an ongoing research focus, with efforts to address these limitations through improved coatings and electrode designs. Researchers are actively working on developing coatings and improved electrode designs to reduce droplet evaporation and prevent fouling, thereby enhancing the long-term reliability of the devices. It's like refining a high-tech car. Even though it performs exceptionally well, engineers must continually work on reducing wear and tear on its components to ensure it remains reliable over long journeys. The future of digital microfluidics is promising, with ongoing innovations aimed at further increasing automation and integration in laboratory workflows. Advancements are expected to improve device robustness, scalability, and user-friendliness, with integration of artificial intelligence and machine learning likely to enhance data analysis and decision-making capability. These developments will further streamline complex laboratory procedures, reducing time and costs associated with research and diagnostics, and ultimately revolutionizing lab automation by enabling smarter, faster, and more efficient workflows. Future digital microfluidic systems are being developed that use AI to optimize droplet handling in real time, adjusting protocols automatically to ensure the best possible experimental outcomes. Imagine upgrading from a traditional manually operated factory to a fully automated smart factory where machines communicate and adjust operations in real time to maximize efficiency and output. Digital microfluidics is transforming laboratory automation by harnessing the power of reconfigurable droplet platforms to carry out complex workflows with unprecedented precision and flexibility. At its core, this technology uses electrical signals to manipulate tiny droplets on an array of electrodes. These droplets can be precisely moved, merged, split, and mixed on a flat surface, enabling a wide range of chemical and biological reactions to occur in a controlled automation. This reconfigurability means that a single device can perform multiple tasks, adapting dynamically to the needs of different experiments or diagnostic tests. One of the key advantages of digital microfluidics is its ability to miniaturize and integrate multiple laboratory processes onto a single chip. Traditional lab workflows often require a series of manual steps, from sample preparation and reagent mixing to incubation and detection. With digital microfluidic platforms, these steps can be automated in a seamless sequence. For instance, a droplet containing a biological sample can be directed to merge with another droplet holding a specific reagent and then transported to a sensing area for analysis, all without human intervention. This not only speeds up the workflow, but also reduces the potential for error and contamination, which is especially critical in sensitive diagnostic and research applications. The versatility of reconfigurable droplet platforms is further enhanced by their programmability. Scientists can design and execute complex protocols by simply reconfiguring the electrode patterns and controlling the timing and voltage applied to each droplet. This means that a single digital microfluidic device can be repurposed for a variety of assays, ranging from DNA amplification and sequencing to chemical synthesis and drug screening. Imagine a research lab where one chip can switch between performing polymerase chain reactions, PCR, one moment, and conducting enzyme assays the next, simply by altering the control software. Such flexibility not only streamlines laboratory operations, but also paves the way for highly customized workflows in precision medicine and high-throughput screening. In addition to streamlining traditional lab processes, digital microfluidics is pushing the boundaries of what is possible in lab automation. For example, these platforms can be integrated with optical, electrical, and even mass spectrometry sensors, allowing for real-time monitoring and data acquisition directly on the chip. This integration facilitates immediate feedback and rapid decision-making, crucial in clinical settings where time is of the essence. Could this technology eventually lead to fully autonomous laboratories where experiments are conducted, analyzed, and adjusted in real time without human oversight? Digital microfluidics for lab automation represents a significant leap forward in how we approach laboratory workflows. Its ability to automate complex procedures in a miniaturized, reconfigurable format not only accelerates scientific discovery, but also enhances the reproducibility and accuracy of experiments. As researchers continue to refine these platforms, we can look forward to a future where lab automation becomes more accessible, efficient, and adaptable, ultimately transforming research, diagnostics, and the development of new therapeutics.